Good afternoon. I thought I would do something slightly different this afternoon, and rather than just pitch Immunocore's platform technology, or even our lead drug, I thought I would focus more on how we've taken our lead drug safely into the clinic in both the UK and the US without any animal toxicity testing whatsoever. So, Immunocore's drugs are based on T-cell receptors. These have a significant advantage over antibody-based technologies because they can be used to target absolutely any disease-specific protein, even if that protein is intracellular, where an antibody physically cannot get to it. How does this magic work? Every nucleated cell in your body takes all the different types of proteins it's making, intracellular, secreted, membrane, and it breaks them down into short peptide fragments. Those fragments that meet very specific length and sequence criteria are picked up by a protein complex called human leukocyte antigen, and then the HLA peptide complex is exported to the cell surface, where effectively it acts as a flag of all of the metabolic activity that's going on inside that cell. A passing T-cell wanders along, and with its T-cell receptor, it scans all of the HLA peptides that are being presented to it. If it binds to that epitope and recognises it, that epitope shouldn't be there, there's something wrong with the cell, and the T-cell will kill it. So basically, we use these T-cell receptors as a new form of targeting moiety that just give us an advantage over other scaffold antibody technologies. So what do our drugs actually look like? We take those T-cell receptors and we turn them into soluble biologic drugs that are bispecific. So we take a highly engineered T-cell receptor and we use that as a targeting moiety. To give it a biological function, we then fuse it to an anti-CD3 scFv fragment, and we use this to redirect the body's T-cells against your drug-targeted cell. A couple of key points. In order to make the T-cell receptor a true targeting system, we need to increase its affinity several millionfold, and we do that by phage display, affinity maturation.